Hello everyone, Biochemistry channel is here with the topic titled Amino Acids, Peptides and Proteins-III. Please visit biochemia.web website for a more comprehensive document of the subject in PDF format. You can find the link in the description. And please do not forget to subscribe, like the video and turn on notifications before starting. I wish it will be useful. Let's get started. The contents of this topic are nucleoproteins. Actually, we mainly talk about nucleic acid, a component of nucleoprotein. And then chromoproteins, porphyrins, hemoglobin, myoglobin, and we will talk about the Icterus, the Jaundice. At the end, I will give you some information about major proteins of organism, fluids and tissues, mainly fluids. I will talk about the blood proteins. The first title is nucleic acids. Nucleic acids are the prosthetic group of nucleoproteins, molecules that have hereditary properties, carry genes, and are key to protein biosynthesis. They were first isolated from the cell nucleus, hence the name nuclein or the nuclein substance. Nucleic acids are involved in every specific biological event, including the breakdown of the cell. There are two compounds in the structure of nucleic. These are the constitutional blocks of nucleic acid, and they are nitrogenous base, pentose, and phosphate. So a nitrogenous base plus a pentose forms a nucleotide. By adding a phosphate, we call it a nucleotide. A number of nucleotides forms links to form nucleic acid. So this is the terminology. A nucleotide means 1 mole of nitrogen base plus 1 mole of pentose and 1 mole of phosphate. And the moiety of nitrogenous base and pentose is called nucleoside. And number of nucleotides links to form nucleic acid. So nucleic acids are the polymers of mononucleotides. Here you see the molecular structure of a nucleotide. Nucleotide having a nitrogenous base, and here in the example it is adenine, as I will talk about nitrogenous bases on the following slide, a sugar moiety and a phosphate group. In the structure of a nucleotide, pentose is in the middle. It is attached to the nitrogenous base with the first carbon, as you see here, and the phosphate to the fifth carbon. And the third carbon of the sugar binds to the next nucleotide or links with the next nucleotide, the phosphate group of the next nucleotide. So the sugar actually here provides a kind of bridge to hold nitrogenous base, the one hand, and on the other hand holds the phosphate group, and also the third hand holds another nucleotide here. And pentose is Deoxyribose or Ribose. It depends on the type of nucleotide. Actually, correction depends on the type of nucleic. In DNA, Deoxyribose. In RNA, Ribose. And here in the given example, this nucleotide is a part of the Deoxyribonucleic Acid, the DNA. Because here at the second carbon of the pentose sugar, there is an hydrogen atom, not the alcohol atom. So this is the deoxy sugar, my dear friend. And here you see the nitrogenous bases. And there are two classes of nitrogenous bases in a nucleotide. We call them the purine and pyrimidine bases. And also there are some rare different type of bases. Methyl, dimethyl, and N-methyl derivatives of these bases. And the purine bases are adenine, abbreviated A, and guanine, abbreviated as G. And the pyrimidine bases are uracil, the U, thymine T, and cytosine C. And as you see here, purine bases are two ring bases, or they carry a two ring system. But in pyrimidines, as you see here, they carry only one ring. And in purines, they carry 4 nitrogens, and pyrimidines have 2 nitrogens in the ring structure. Pyrimidines are heterocyclic aromatic compounds. And as you see, we can easily guess it's a derivative, a kind of derivative of benzene ring. But in the purine, they carry here the heterocyclic aromatic ring plus a fused imidazole ring. This is the second ring in purine bases. And the catabolic end products are also different. For the purines, the catabolic end product is uric acid. But the pyrimidines, the catabolic end products are beta amino acids, carbon dioxide, and ammonia. Let's look at the differences between the pyrimidine base. And as you see, the thymine pyrimidine base carry a methyl group at the fifth carbon. But in the uracil, as you see, there is no methyl group at the fifth carbon atom. In the cytosine, at the fourth carbon atom, it carries an amino group, as you see here. These are the difference between pyrimidine base. Another difference is cytosine is a pyrimidine base of both DNA and RNA. But for the uracil and thymine, it is unique because thymine is a base of only DNA. However, uracil is only for RNA. This is a big difference. The structural difference of these pyrimidine bases is responsible for the various roles, and especially for the thymine in DNA and uracil in RNA. In DNA, thymine pairs with adenine, while uracil is found in RNA and pairs with adenine. Both thymine and uracil are important for processing and transferring genetic information. Cytosine has an amino group, what I said, at the fourth carbon atom. It's found in both DNA and RNA. What about the purines? The main difference between adenine and guanine is that adenine contains an amino group on carbon 6 and an additional double bond between the first nitrogen and the carbon 6 in the pyrimidine ring. So this means pyrimidine ring, also we call it pyrimidine ring because it's the same with the pyrimidine. The difference is in they carry a fused imidazole ring. While in guanine, it contains an amino group on carbon 2 and the carbonyl group on carbon 6 in the pyrimidine ring. As you see here, the carbonyl group at the 6th carbon. So these differences ensure that DNA, RNA function correctly to store and transmit genetic information correctly. Cells can synthesize purine and pyrimidine bases. Purines and pyrimidines are the building blocks of nucleic, which are vital for the storage and transmission of genetic information. Every cell has the capacity to build these molecules through specific biochemical pathways. Synthesizing these bases is vital for cell function and division, as they are needed to create the genetic material for new cells. Cells can recycle these bases from old, broken down molecules or synthesize them from scratch from smaller components such as amino acids, carbon dioxide, and nitrogen. Maintaining the balance between synthesizing and recycling these bases is crucial for proper cell function and genetic integrity. Distribution of these pathways can lead to various health problems, genetic disorders, and even cancer. The components used in the synthesis of purine and pyrimidine bases are, what are they? For the synthesis of purine bases, they are glycine, aspartate, glutamine. These are the amino acids. These are the nitrogen providers. Also, these are the providers of backbone of these ring structures or the ring structures of these bases, formyl groups, and carbon dioxide. For the synthesis of pyrimidine bases, aspartate, glutamine, and carbon dioxide is required. And these components are combined through a complex series of enzymatic reactions with the help of the energy carrier molecules such as ATP and NADPH. The NADPH is nicotinamide, adenine, the nucleotide phosphate, an electron carrier in the biosynthetic pathway. The purine and pyrimidine synthesis pathways are energy-intensive processes, and cells expend significant amounts of energy on these processes. Therefore, under certain conditions, cells can save energy by obtaining these bases from nutrients known as the salvage pathway. Deoxyribose and ribose, the sugar molecule nucleotides as well as nucleic. Nucleic acids, DNA and RNA, are named after the pentose in their structure. If deoxyribose is present, it is called DNA, deoxyribonucleic acid. If ribose is present, it is called RNA, ribonucleic. The structures of these molecules consist of the sugars, consist of the pentoses, monosaccharides having 5 carbon atom and aldehyde group, together with bases and phosphate group. One of the main differences between DNA, RNA lies in their molecular composition. And let's look at the difference of molecules of deoxyribose and ribose. And in DNA, which contains the deoxyribose, that one, oxy sugar, as you see, at the second carbon of the carbon, there is no alcoholic group. It is reduced. Ribose, this is the common form in nature, having an alcoholic group at the second carbon, it is reduced to hydrogen. And this reaction is catalyzed by ribonucleotide reductase enzyme. Ribonucleotide reductase enzyme, an enzyme that catalyzes the formation of deoxyribonucleotides from ribonucleotides. It catalyzes this formation by removing the second hydroxyl group of the ribose ring of nucleoside, the phosphate. Thus, the reduction produces deoxyribonucleotides, and then using the synthesis of DNA. There are also free nucleotides in body, which they possess very important functions. Nucleotides are found in the structure of nucleic acid as well as in free form in tissue. There are many, and most of these have catalytic functions and work together with enzymes. In other words, they are the coenzymes of the enzyme. And according to nomenclature, the nitrogenous base plus pentose gives nucleosides, such as adenine plus ribose, adenose. And if we add a phosphate moiety, it will be adenosine monophosphate. Phosphate moiety, adenosine diphosphate, and if there are three phosphate groups, it is called adenosine triphosphate. And in this table, you see the naming of a nitrogenous base, its nucleoside and nucleotide having only one phosphate moiety and its symbol. And then example, for example, adenine is nucleoside. This means adenine plus ribose is called the adenosine. If one phosphate moiety is attached to the ribose, it will be adenosine monophosphate or adenylic acid. So here the oleic acid refers the monophosphate. So instead of saying adenosine monophosphate, which is the common way to express it, we can also say as an alternate adenylic acid, guanilic, delic, and pyrimidinic acid for the pyrimidine monophosphate. And here are some important free nucleotides and the common ones, especially we need them in metabolism, and we will express them too many times in the biochemistry 2 core. And AMP, ADP, and ATP, they are the free nucleotides in energy metabolism. NAD, NADP, MN and FAD also, they are the electron carriers in energy metabolism, or they are electron carriers, or they are reducing equivalence in oxidoreductase class of enzymes, or in redox reaction. In UDP-G, which means within the phosphate glucose, it's a substrate carrier in glycogen synthesis. And the coenzyme A, my dear friends, is critical in the transport of acid groups, particularly in the citric acid. These molecules play fundamental roles in cellular energy production, regulation of metabolic pathways, and numerous biochemical reactions. And common characteristics of these compounds include energy transport, electron transfer, and to be a coenzyme of certain enzymes. And interestingly, they are especially the coenzymes of the key enzymes in metabolism, or they are the coenzymes of the regulatory enzymes that we will study in biochemistry 2. And this is a molecular pattern of adenosine mono D and triphosphate. Actually, it's a triphosphate. As you see, there are three phosphate groups, and they are attached with phosphoanhydride bond, which store the energy when in metabolism. And as you see, it's a ribose here because the second carbon have an alcoholic group, and the base here is adenine. And the nitrogenous base plus the ribose, this moiety is adenosine, and with one phosphate, it is called adenosine monophosphate or adenylic acid. The second phosphate moiety does the name adenosine diphosphate, and the third phosphate moiety does the name adenosine triphosphate. This is the name, a simple naming. What are the functions of nucleic acids? Nucleic acids are biomolecules found in living cells that play critical roles in the energy storage, expression, and transmission of genetic information. And we know very well that there are two main types, DNA and RNA. DNA carries the genetic instructions of living organisms. These instructions are essential for the development, function, and reproduction of the organism. The information in DNA is translated into proteins via RNA, a process called gene expression. There are various forms of RNA, and each one performs different functions. For example, mRNA, the messenger RNA, carries genetic information from DNA to ribosome. tRNA, transfer RNA, brings amino acids to the ribosome and is involved in protein synthesis. Ribosomal RNA forms the structure of the ribosome, plays a central role in protein synthesis. These structures and functions of nucleic acids are fundamental to the functioning of living cells and the transmission of genetic information from generation to generation. The discovery and study of these molecules has revolutionized the field of biology and medicine. Let's look at the DNA to talk more on DNA, deoxyribonucleic acid. DNA is made up of units called nucleotides. We know each nucleotide contains a phosphate group, a sugar moiety, which is deoxyribose, and one of four bases, adenine, thymine, guanine, and cytosine. These nucleotides are linked together to form a double helix. In a DNA, adenine and thymine or guanine and cytosine are always equimolar, but the ratio of adenine plus thymine and guanine plus cytosine can vary. Let's look at the molecular structures of nucleic. And as in the proteins, they have a primary structure, and the primary structure of nucleic acid is the linear arrangement of nucleotides. There are secondary structure of nucleic acid, and as you see here, an example for RNA. A secondary structure encompasses small local folding motifs, helices, stem loops beside the canal. And for the DNA, actually, this secondary structure refers the double polynucleotide chain pairing of nitrogenous bases between two polynucleotide chain refers to secondary structure of DNA. And also there are tertiary structure, refers to the three-dimensional folded form of the nucleic acid molecule. And for the DNA, helical structure, folded helical structures, and we will discuss the DNA, A DNA, B DNA, and Z DNA. And also this happens in the RNA molecule. And also there are quaternary structures. And the quaternary structure generally donates three-dimensional interactions between multiple subunits, such as with histone protein, or the protein nucleic acid interactions provide the quaternary structure. And the base pairing is so important in both in DNA and also in the motif structure of the RNA, as you see here. The guanine, cytosine, and uracil adenine forms hydrogen. And between two polynucleotide chains, as you see here, also adenine, thymine, and guanine, cytosine forms the pairs. Or they pair, and they link with hydrogen. And this specific pairing ensures the accuracy of the molecule. And for the DNA, ensures accurate replication of DNA and the integrity of genetic information. The structure of DNA is crucial for the storage and transmission of genetic information, enabling the compact packaging of vast amounts of genetic data within the cell nucleus. And genetic information in DNA expressed through genes. Each gene contains instructions for the synthesis of a specific protein or RNA molecule. Gene expression involves the processes of transcription of DNA to RNA and subsequent translation to proteins. These processes are vital for the development, function, and reproduction of living organisms. So, the detailed structure of DNA is a fundamental focus in biological research, leading to groundbreaking advancements in genetics, molecular biology, medicine, and biotechnology. In the previous slide, I said there are three forms of DNA: A DNA, B DNA, and Z DNA. These are the three distinct conformations of DNA, and these structures result in different physical characteristics of DNA's double helix, each demonstrating how DNA behaves under varying environmental conditions. And in the B DNA, here in the B DNA, identified by Watson and Crick, it is the most common form of DNA found in cells, and it has a right-handed helical structure with about 10.5 base pairs for a complete turn. The base pairs are nearly perpendicular to the axis of the helix. Look at here. This structure is most stable under normal hydration levels and ambient conditions within the cell. The universally common form in all living organisms is the right-handed double helix known as pDNA. Look at the aDNA. The difference. aDNA possesses a tighter right-handed helical structure compared to pDNA and is typically observed under the hydrated condition. The helix of aDNA is broader and shorter, containing about 11 base pairs per complete turn. The base pairs are inclined at a more accurate angle to the helix axis. It forms under low humidity conditions or in the presence of certain solvents and is rarely seen in normal cellular conditions. What about zDNA? zDNA featuring a left-handed helical structure, and it's a less common form of DNA. It has a zigzag shape, as you see, and contains 12 base pairs per turn. The base pairs are aligned with a narrower helical turn. It generally forms under high salt concentrations or in certain base sequences. This structure is biologically active and may play a role in the regulation of gene expression. Understanding all three DNA conformations is crucial in elucidating how DNA adapts to environmental conditions and can exhibit different structural properties under various physical circumstances. These structures hold significance in genetic research as well as in studies exploring DNA's chemical and physical properties. DNA is one of the basic building blocks of living cells and performs a number of vital tasks. These are storage of genetic information, transfer of genetic information, protein synthesis, regulation of cell functions, cell repair and division, evolutionary change. And the roles of DNA are fundamental to the biological structure and functions of living organisms. And proper functioning of DNA is crucial for understanding health and disease, treating genetic disorders, and in biotechnological applications. In cell division, the genetic elements are transferred to the offspring completely and unchanged. For a new DNA molecule to be formed, which is called DNA replication, the double helix tertiary structure must be broken and the secondary structure must be formed. And the separated DNA strands form a mold for the new DNA strands to be formed. In the face of this mold, only a DNA strand similar to the structure of the partner strand from which this strand was separated can form. Because the nitrogenous bases in the DNA strand, which we call the template, can only match with nitrogenous bases that match their sequence. Since the strands formed here are completely similar to the parental double strand in terms of base sequence, and each of the double strands contains a parental strand, replication is called semi-conservative replication. The three nitrogenous bases on top of each other in the DNA molecule encode an amino acid and are called codon. And what is meant here is the DNA codon. There is also the mRNA codon and an anticodon in tRNA. And here you see the codon and the representative amino acid they code. And as you see, some amino acids, for example here, tryptophan, there is only one type of codon. But for tyrosine, there are two types. For serine, look at here, 1, 2, 3, 4, 5, 6. There are 6 different codons encode the amino acid. And for each protein synthesis, there is a starting codon, and each protein synthesis starts with the methionine. And remember, methionine is an essential amino acid, and the starting codon is adenine, thymine, guanine. And also there is a stop codon, which not code any amino acid, and these are the stop codons. And as you see, all codes for amino acids are known, and these codes are used to synthesize the messenger RNA, which means these codes are delivered to the mRNA, because mRNA has the codon for each amino acid. But instead of thymine, mRNA carry uracil. So you can change these codes, you can change the T abbreviations of these codes to the U abbreviation to represent the uracil mRNA. And for all, there's an anticodon transfer RNA. And as you see here, if there are many various codons for an amino acid, for an individual amino acid, this means also there are many transfer RNA. It is time to speak ribonucleic acid. RNA is also composed of nucleotides. In order for RNAs to be formed, primary DNAs are required. A question, and the answer, is the RNA structure different from the DNA structure? Yes. Instead of deoxyribose, it carries ribose. Uracil is found instead of thymine, and it's not a double. one, as you see here, one strand, but it has tertiary strand. There are three main types of RNA. These are messenger RNA, ribosomal RNA, rRNA, transfer RNA, tRNA. And messenger RNA carries genetic information from DNA to the ribosome. Ribosomal RNA forms the structure of the ribosome and plays a central role in the proteins. And tRNA brings amino acids to the ribosome and plays a role in protein synthesis. Actually, all plays a role in protein synthesis. And also we can say all simultaneously and highly organized work together for the protein synthesis. Let's look at the difference between DNA and RNA. We know one of the differences about monosaccharide. They have in RNA or ribose, but in DNA, deoxyribose. In DNA, one of the pyrimidine base is thymine. Instead of thymine, RNA carry uracil. This is the second. And the third one is there are two polynucleotide chain in DNA or two strands, but in RNA, one single strand. Let's talk about the central dogma of molecular biology. The central dogma of molecular biology refers to the unidirectional flow of genetic information from DNA to RNA and RNA to protein. And this dogma elucidates the fundamental principles of storage, expression, and utilization of genetic information and plays a crucial role in understanding the functioning of all living cells. And this theory was developed in 1958 by Francis Crick, and his version was more global, incorporating the concept that information does not flow from proteins to nucleic acids. It's a unidirectional flow of genetic information. However, following developments in molecular biology have revealed there are certain exceptions, and the notable example is Purines, which are infectious proteins that replicate without passing through DNA or RNA intermediate. And Purines are responsible for rare but devastating neurological diseases such as Creutzfeldt-Jakob, characterized by uniform fatal degeneration of the nervous system. Another example is the process of reverse transcription in RNA viruses, such as retrovirus, coronavirus, the process reverse transcription. That kind of viruses carry an enzyme, reverse transcriptase enzyme, which facilitates the flow of information from RNA to DNA. Therefore, while the central dogma provides a general framework for the flow of genetic information in living systems, it is necessary to acknowledge the existence of more complex and diverse mechanisms. And the flow of genetic information from DNA to RNA is called transcription, RNA to protein, translation. Let's talk about transcription and translation. Transcription and translation are two fundamental processes in protein synthesis and play central roles in the transfer of genetic information from DNA to protein. Transcription is the process by which the genetic information in DNA is copied into RNA, into messenger RNA. And transcription involves three stages: initiation, elongation, and termination. At the end, an mRNA, messenger RNA molecule containing the genetic information from DNA is formed. This messenger RNA is then transported to the cell cytoplasm to participate in translation. Translation is the process by which the genetic information in messenger RNA is translated into protein. In this process, the amino acids specified by the codons in the messenger RNA are sequentially combined to form a protein. And like transcription, translation consists of initiation, elongation, and termination stages. The outcome of translation is the production of a protein based on the genetic information in mRNA. And these proteins meet various structural and functional needs of the cell. These steps in the expression of genetic information enable cells to produce the correct proteins at the right time, supporting the growth, development, repair, and various biological functions of living organisms. These Processes are fundamental mechanisms of molecular biology and provide essential insights for understanding genetic diseases, gene therapy, and biotechnological applications. The messenger RNA, one type of RNA. DNAs determine the sequence of amino acids in the structure of protein. All three nitrogenous bases in the polynucleotide chain of a DNA encode an amino acid. Here, the order of the nitrogen bases in the nucleus of the DNA is genetic. The first step in protein biosynthesis is that the genetic information in DNA is transported to ribosomes where protein synthesis occurs. This is done by messenger RNAs, and it is called transcription. An RNA template is formed that represents the genetic information and DNA portion necessary for protein synthesis. It passes into the cytoplasm and binds to the ribosome. The amino acid code is valid for all forms of life, including viruses that have been researched up to now. The genetic code of DNA is carried by the base sequence of RNA, and the base sequence of the mRNA determines the amino acid sequence of protein. rRNA, ribosomal RNA. Ribosomal RNA is a type of RNA found in the structure of ribosomes and plays a central role in protein synthesis. Ribosomal RNA functions as both structural and catalytic components of the ribosome and is vital for protein production in the cell. Ribosomes are complex molecular machines essential for protein synthesis and are located in the cytoplasm of the cell. In eukaryotic and prokaryotic cells, ribosomes can be of different size and structure, but in both cases, ribosomes consist of two main units, large and small subunits. And don't forget, the ribosome is a really complex cellular mesh, and it is largely composed of specialized RNA known as ribosomal RNA and dozens of different proteins. Ribosomal proteins and ribosomal RNAs are organized into two separate ribosomal parts of different sizes, large and small subunits of the ribosome. Ribosomes consist of two subunits. Here, the blue one represents the smaller subunit, and the red one represents the larger subunit. In eukaryotic ribosomes, they are referred as ATS. And here S represents the sedimation velocity or the sedimation rate. But in prokaryotes, they are called 70S. So they have different sedimation rate. And both units composed of different type of RNA. And for example, in the eukaryotes, the large subunit is then 60S and the smaller subunit is 40S. In prokaryotes, the large unit 50S and the smaller unit 30S. They composed of different proteins and RNAs. For example, in large subunit of eukaryotes, the 60S, it contains type of 28S, 5.8S, and 5S ribosomal RNA, different, along with approximately 49 proteins. The large subunit of ribosome includes catalytic functions necessary for the formation of peptide. In the smaller subunit, it facilitates the binding of messenger RNA and the codon-anticodon pair. Transfer RNA, tRNA. RNA molecules capable of combining with special amino acids and transporting them to ribosomes. These are transfer RNA. They have three important features. They can covalently bind to a special amino acid. They have a triple code within the structure that complements the amino acid in the mRNA codon, therefore called an anticodon. So this is the anticodon moiety here, and here is the moiety which covalently bind the special amino acid regarding to the anticodon. The combination of first two of the bases representing the codon of messenger RNA and the anticodon of transfer RNA occurs according to the principle in DNA. Transfer RNAs are small and stable RNA molecules, typically comprising 70 to 90 nucleotides. And as you see, its structure often described as a clover leaf shape. This structure results from the unique folding of the transfer RNA molecule and the stabilization by hydrogen bonds. And here you see all major types of ribonucleic acid. Ribosomal RNA, here the mRNA and the transfer RNA. And the diversity of transfer RNA varies depending on the organism's genome and its environmental content. In many eukaryotic organisms, including humans, there are hundreds of different transfer RNA genes. And these genes typically produce tRNA molecules specific to certain amino acids and corresponding to the three-letter codons of the genetics. As a result, in many organisms, including humans, there are hundreds of different transfer RNA molecules, each coding for different amino acids and recognizing different codons. However, this number can vary depending on the genomic diversity and biological needs of the organism. And my dear friends, we also know different types of RNA molecules exist in the cell. So these are the major ones, but also there are the smaller ones, which have the catalytic activity. An example is snRNA, different type of RNA, small nuclear RNA, particularly plays a role in RNA processing, such as maturation of messenger RNA. snRNA is a component of complexes called spliceosomes and facilitates the removal of introns from messenger RNA. Another example is miRNA, which is microRNA. MicroRNA plays a significant role in regulating gene expression. It regulates gene expression by causing the degradation of target messenger RNA or by inhibiting its translation. This regulation impacts numerous biological processes, including cell differentiation, growth, and apoptosis. There are certainly other types of RNA as well, which you can explore them from the web. Each type of RNA plays unique and critical roles in the regulation and expression of cellular functions and genetic information. And this diversity enables cells to perform complex biological functions. And we'll finish the nucleic acids. Chromoproteins. Chromoproteins are conjugated proteins whose prosthetic group is colored substances such as porphyrin. Chromoproteins are proteins that contain one or more colored prosthetic group. In general, we call them chromophores within their structure. And these chromophores are colored molecules that confer specific characteristics to the protein and play a significant role in the protein's biological function. Chromoproteins play roles in many fundamental functions of living organisms. These include functions such as respiration, photosynthesis in plants, light detection, and energy production. An example is hemoglobin and respiratory enzymes. They are chromoproteins whose prosthetic group is heme. Heme is a prosthetic group of these molecules. Another example is flavoprotein, with riboflavin as a prosthetic group, and proteinous substances in hair and wool with melanin as a prosthetic group are also chromoproteins. Catalase enzyme is a chromoprotein whose prosthetic group is heme, another molecule carrying heme. And by the way, catalase, hemoglobin, and respiratory enzymes are also metalloproteins because in the structure of heme, iron is carried. Chromoproteins carrying heme as a prosthetic group are called hemoproteins. So hemoglobin, myoglobin, cytochromes, and peroxidases are hemoproteins. And the study of chromoproteins is a fundamental topic in biochemistry. Understanding the structure and functions of these proteins is important for a better comprehension of health and disease mechanisms. Chiral ring, here as you see, a simple ring structure, if I remember, is the first and simplest basic part of the porphyrin. If propionic acid, as you see, this is a kind of modification of chiral ring, so it may be propionic acid, acetic acid, or side chains such as methyl, ethyl, guanine are added to the chiral ring. A structure called porphobilinogen (PBG) is formed, and the porphobilinogen is called the monochiral, one single unit chiral. It is the precursor of porphyrin. Porphyrins are formed when four porphyrinogen rings, and you see here there are four porphyrinogen rings, are linked together by the methane bridge. And here these are the methane bridge, my dear friend. Porphyrins are also defined as derivatives of the porphyrin precursor, which is a closed structure consisting of four chiral rings without side chains. Porphyrins are cyclic and complex organic molecules, usually bound to metal ions to form metalloporphyrin. These molecules play important roles in biological systems and are particularly critical in the processes of respiration and photosynthesis. And here is some basic information about the porphyrins and additional. As I said, they have metal binding capacity. Porphyrins usually have a metal ion in the center. The center is here. And this metal can usually be iron, as seen in the hemoglobin, myoglobin, maybe magnesium, zinc, cobalt, and copper. And the cobalt, for example, in cobalamin, vitamin B12. And the metal ion is coordinately bound to the porphyrin ring. And the ring having iron is called heme. If we have an iron at the ring, a coordinated complex of four single bonds, and one attached to the histidine and the one empty moiety for molecular oxygen, it is called heme. It's a very critical part of the hemoglobin and myoglobin. It gives the functionality, and the heme is an iron containing porphyrin, and the proteins having heme are called hemoglobin. It has a critical role in the transport and storage of oxygen. Another example is cytochrome. Cytochromes also carry heme. They are involved in the electron transport chain and also some redox enzymes or some redox reactions. So they are very critical molecules. Let's talk about hemoglobin. But don't forget, also we talk about hemoglobin in the topic part 2, in the amino acids, peptides, and proteins part 2. Hemoglobin is a chromoprotein carrying heme as a prosthetic group. It is formed by the binding of four heme groups. So this is very important. There are four heme groups parallel to each other and perpendicular to the peptide chain with globin. Hemoglobin is rich in histidine and is a good buffer due to its excess in the blood. Hemoglobin's most important task is to combine with atmospheric oxygen in the lungs by forming oxyhemoglobin and transport oxygen to tissues. And most specifically, hemoglobin synthesizes occurs in the precursor cells of the erythrocyte, known as erythroblasts, located in the bone marrow. And the process begins in the early stages of red blood cell formation in the bone marrow. As erythroblasts mature, they progressively accumulate hemoglobin. And after these cells transform into reticulocytes, an immature form of red blood cells, they enter the bloodstream. Within a few days, they mature into fully developed erythrocytes carrying hemoglobin as their primary component. And here you see the heme group together with the hemoglobin. And the heme group found in critical proteins such as hemoglobin, myoglobin, containing porphyrin. And as you see in the hemoglobin, there are four heme groups, which means the hemoglobin binds 4 moles of oxygen. And there are different types of hemoglobin in humans and also in other living. And the most common types of hemoglobin in humans, my dear friends, are hemoglobin A, hemoglobin A2, hemoglobin F, and also there are other types of hemoglobin such as hemoglobin S. You may remember from the second part I talked about, hemoglobin C, and the other ones. So these are the normal hemoglobins, but these are the abnormal hemoglobin. And hemoglobin A. So what is the difference between hemoglobin A, A2, and F? And hemoglobin A is the most common type of hemoglobin in adults, accounting in 97% of hemoglobin is a hemoglobin A in an adult. And this hemoglobin is the hemoglobin carrying 2 alpha, and 2 beta chains, as you see in the picture here, illustration. In hemoglobin A2 is present in liver, about only 2%. Only 2%. Also, it's one of the globin is different. In hemoglobin A2, there are 2 alpha and 2 delta. So one globin chain is different, not better. And hemoglobin A2 levels are an important diagnostic indicator for some blood disease, especially thalassemia. So what is hemoglobin F? F here represents fetal. This is fetal hemoglobin, the predominant type of hemoglobin in fetus and in the first month after birth. And it carries a 2-alpha and 2-gamma chain. As you see in all normal hemoglobins, they carry 2-alpha, but they differ in the second type of the chain. A, beta. A2, it's delta. In fetal hemoglobin, it's a gamma globin. In the hemoglobin, the fetal hemoglobin is so crucial for the fetus because fetal hemoglobin can transport oxygen more efficiently than the other one, which is necessary to meet the oxygen demand of the fetus. So hemoglobin S is seen in sickle cell. It is called sickle cell hemoglobin, and it's a type of hemoglobin characterized by a single amino acid chain change in the beta globin chain and causes sickle cell anemia. Hemoglobin C, another type characterized by another amino acid chain in the beta globin chain and causes hemoglobin C disease. And also there are some other variants such as hemoglobin E and hemoglobin D. And hemoglobin types, as you see, vary due to genetic variations and mutation, and these differences are important in the diagnosis and treatment of various hematological conditions. Hemoglobin types differ in their oxygen carrying capacity and oxygen binding properties, which in turn affect the way the body meets its oxygen requirement. There are some basic definitions that we should know, my friends. If hemoglobin combines with oxygen, we call it oxyhemoglobin. If it combines with carbon dioxide, we call it carbaminohemoglobin. If it combines with carbon dioxide, it's called carboxyhemoglobin. So here, please be very careful. Don't think the carboxy represents carbon dioxide. It represents carbon monoxide. And carbon monoxide is a poison. It's very dangerous. One carrying carbon dioxide is called carbaminohemoglobin. Be very careful. If iron reacts with oxidizing agents, and by the way, don't forget the iron in hemoglobin is in 2 ionized state. It's a ferrous iron, not the ferric iron. So if iron reacts with oxidizing agents, methemoglobin occurs. And the methemoglobin also a kind of hemoglobin which no oxygen carrying capacity occurs or decreasing dramatically, and it may be life-threatening. If iron can also be replaced by some anions, such as cyanide, to form cyan-methemoglobin. And if oxyhemoglobin is treated with hydrogen sulfur, sulfur-hemoglobin is formed. There is also another type of hemoglobin, which is hemoglobin A type 1C. Hemoglobin A 1C. And this is glycosylated hemoglobin. Hemoglobin can react with glucose in the blood to form hemoglobin A1C. And it is used as an indicator of long-term blood glucose control and is important in the management of diabetes mellitus. And these different forms of hemoglobin are important for understanding how hemoglobin functions in the body and how it interacts with various substances. The properties of each form affect the carrying capacity of hemoglobin and how oxygen is delivered to tissue. Myoglobin. Myoglobin is a protein found in muscle cells, functions to transport oxygen. Myoglobin is composed of a single polypeptide chain, differs from hemoglobin, which consists of 4 polypeptide chains. And myoglobin contains one heme group, a single polypeptide chain and a single heme group, which provides its oxygen binding capacity. And its polypeptide chain contains 153 amino acids. And my dear friends, myoglobin has a higher affinity for oxygen than hemoglobin. And this allows myoglobin to store oxygen more effectively in muscle cells. Myoglobin increases the oxygen storage and transport capacity of animals' muscle tissues, enabling them to survive in various environmental conditions. Myoglobin levels are especially high in animals living in environments where oxygen is limited, such as diving stations. Myoglobin is an important protein that enables muscle tissues to function effectively and meets the oxygen demand for muscles during intense physical activity. Thus, myoglobin plays an important role in both physiological processes and also in the medical diagnosis. Another type, bile pigment. Red blood cells are destroyed after a lifespan of about 125 days, and as a result, the porphyrin ring is opened. An open chain containing 4 pyrrole rings is formed, which are called bile color substances. In tree, we call them bile. All substances included in the group of bile pigments are theoretically considered to derive from a parent Bile substance, also called protobil. The bile pigment formed in the first step in the breakdown of hemoglobin is biliverdin. And bilirubin is formed by the reduction of one of the methylene groups that bind the porphyrin rings in biliverdin to the methylene. So shortly, destroyed, hemoglobin is degraded into the porphyrin ring and the globin. And the globin they hydrolyze to its building blocks amino acids, and the amino acids enter to the amino acid lopide. And iron also is recycled, but the remaining porphyrin ring is not recycled. It is converted to biliverdin and then bilirubin. And this happens at reticular endothelial system. And the bilirubin then transported to the liver. Liver conjugates bilirubin with glucuronate, thus forming bilirubin glucuronate, and then secreted into bile and gastrointestinal tract. And in the gastrointestinal tract, the bilirubin glucuronate is converted by bacteria in the gastrointestinal tract to the stercobilin and urobilin. And urobilin is absorbed via vena porta, delivered to the liver, then enters circulation, blood circulation, and removed body from kidneys. And stercobilin is removed in. So this is the short pathway of bilirubin metabolism. Icterus, jaundice. It's a state of hyperbilirubinemia. Collection of bilirubin, which is one of the color substances of bile, is in plasma is called bilirubinemia. So bilirubinemia is a state of the presence of bilirubin in blood. And if it is removed from the kidney within the urine, we call it bilirubinuria, and it's not a normal situation. And if the bilirubin is accumulated in the skin and the mucous membranes and stain them into a yellow appearance, it's called icterus or jaundice. And depending on the reasons of bilirubinemia, actually hyper, don't forget hyper, because bilirubin always present in blood, but in very low concentration. Excess, which is called hyperbilirubinemia, it causes the icterus or the jaundice. And there are 2 types of icterus. The one is prehepatic icterus. This type of jaundice arises when there is an abrupt increase in erythrocyte rate, leading to an excessive accumulation of bilirubin in the blood. Another one is hepatic icterus or hepatocellular icterus. By the way, turn back to the prehepatic icterus, also called the hemolytic icterus, because the main reason is the excessive lysis of erythrocytes. And let's turn back to hepatic icterus or hepatocellular icterus or jaundice. Hepatic icterus occurs due to reasons such as hepatitis and poison, which impair the liver cell's ability to take up bilirubin from the blood and excrete it into the duct. Post-hepatic icterus, also we call it obstructive, retentional, mechanical. This type of jaundice is the result of a mechanical obstruction of the bile ducts due to inflammation, stones, parasites, tumors, etc. And these reasons lead to the accumulation of bile in liver cells and tissue. So depending on the reason, different type of icterus can be seen. And this information is important in the diagnosis of the icterus, because icterus is not a disease. Icterus is a sign of a disease, and to determine the disease, also diagnose disease or disorder or any impairment in tissues, we should know the differences of prehepatic, hepatic, and posthepatic icterus. And the last title of our topic, major proteins of fluids and tissues in organism. Actually, I will talk about the fluids, but the tissues and the rest, please visit the website biochemistry.vet for the PDF document. Serum and plasma. After blood is collected to a tube containing a suitable anticoagulant to prevent clotting, centrifugation separates the blood into two layers. The shaped elements that settle to the bottom, these are the cells of the blood, and the yellow-pink, actually yellow, liquid that remains on top. Because the pink here, well, maybe the red color represents the lysis. Serum, a clear serum without hemoglobin or without lysis, has a yellow in different tones of yellow. So in general, serum and plasma are both components of blood. But the question is, what is the difference? They differ in their composition and the context in which they are used. So the liquid part of the blood that separates in this way, the way is using an anticoagulant, is plasma. And plasma contains 8% of proteins, plasma proteins, 1% of inorganic salts, and the rest contains substances such as lipids, carbohydrates, amino acids, etc. And the fibrinogen fraction of plasma proteins, which constitutes 8%, also included in the plasma. I can also say plasma is the form circulating in the blood, a fluid part of the blood. And it's very critical to maintain blood pressure and volume, supplying critical proteins for blood clotting and immunity, and carrying electrolytes such as sodium, potassium to our body cells. And also the fibrinogen, which is the member of plasma, is a key protein in the clotting process. So what about the serum? Its constituent is almost similar. How different from the blood? Serum is the liquid that remains after the clotting of. So if we collect a blood sample into a tube not containing an anticoagulant and centrifuge the blood, clotting is completely formed. Again, we obtain the yellow fluid part of the blood. That part is called serum because serum does not contain fibrinogen. And in general, serum does not contain blood clotting factor. And here you see the blood after centrifuge, and at the bottom shaped elements. There are the erythrocytes, the biggest part, red blood cells, and at the here there is a buffy coat, we said, carrying leukocytes and thrombocytes. And this is the fluid part, plasma. Water, protein, lipids, carbohydrates, hormones, electrolytes, etc. It carries many substances. And we have a ratio, hematocrit, HCT. And hematocrit is the percentage by volume of red blood cells in the blood. In other words, it's a ratio of blood cells to the total volume. And as you see here, this is the total volume, and this is red blood cell volume. In the normal hemoglobin, as you see, there is almost a similar ratio. In anemia, the volume of red blood cells decreases, which means hematocrit level decreases. Inversely, if there is a polycythemia, this means excess blood cell, hematocrit value increases. And hematocrit value is a good indicator in assessing the hematological condition. Coagulation of blood. The condition arises due to changes in fibrinogen, a protein found in circulating blood. Fibrous protein. When needed, this fibrinogen transforms into fibrin through a series of reactions, and this event is called coagulation or blood clot. The conversion of fibrinogen to fibrin occurs thanks to an enzyme called thrombin, and this enzyme is not active in circulation. In blood clotting, many factors play a role, primarily including calcium. But which form of calcium? The ionized calcium. Free calcium, or the physiological active calcium. Blood clotting is not a simple event, but a complex process with controlled mechanisms. It is referred to as the coagulation cascade, and there are intrinsic and extrinsic factors in the coagulation cascade. And the goal of this cascade is to activate thrombin, thus converting fibrinogen into fibrin. And the platelets are the cellular elements in coagulation. When blood is taken from circulating blood, these platelets adhere to the walls of the tube, or in the case of bleeding, to the edge of the wound, and they disintegrate. And this disintegration releases a substance called thromboplastin. And thromboplastin converts prothrombin, an inactive form of thrombin found in circulating blood, into thrombin. And thrombin then facilitates the conversion of fibrinogen into fibrin, resulting in the clotting of blood. And here you see the two blood volumes, which belongs to the canine and the feline. And here you see the thrombocytes or platelets of the feline and the canine. What are the plasma and serum proteins? Actually, here there was a critical difference. If we have a plasma, we can talk about the fibrinogen because serum does not contain fibrinogen. For the albumin and globulins, we can talk about. This means if we want to measure the fibrinogen concentration in an individual, we must collect plasma. If we collect serum and try to measure the fibrinogen, we will get erroneous results, maybe no results. So be careful. And fibrinogen, a fibrous protein, and it's a component of coagulation cascade. And albumin, a globular protein, globulin also a globular protein. And albumin is important in osmotic pressure and also is a transport protein. And for the globulins, there are different types: alpha globulins, beta globulins, and gamma globulins. And the gamma globulins want the immunoglobulin. As immunoglobulin G, IgG, immunoglobulin IgG, IgE, IgM, and the other ones. And alpha and the beta globulins can give examples such as, for example, alpha-1 antitrypsin. And also there are subtypes such as alpha-1, alpha-2 globulins or beta-1, beta-2 globulins. And there are too many proteins in the family of globulins, which they have various functions. And my dear friends, this is the end of the topic. And here is the references for you to study. And the next topic is enzyme. It will be the last topic of Biochemistry 1. And for more about biochemistry, clinical biochemistry, and laboratory science, you can visit the website biochemia.vet, and also you can follow the Instagram account. Do not forget to subscribe to the YouTube channel, like the video, and turn on notifications to be informed about news. See you in our next one. Goodbye. Stay safe, my dear friends.